Hey, Hammy here. Going to talk today about kind of an overview or introduction for AP biology on cell communication. I've adapted these slides and this lecture series from Campbell Biology 9th edition, chapter 11, and I'll be using their slideshow, which I've modified slightly and be using for my videos here on this series on cell communication. Okay, this first part, we're just going to kind of look at an overview of cellular messaging. In the animal world and plant world, cellular world, I should say, cell-to-cell communication is essential. Cells need to be able to talk to cells. Tissues in one part of an organism need to be able to talk to cells in another part of the organism. Or if you're unicellular, a prokaryote, they have to have a way of communicating with each other, sending messages. Biologists have discovered some universal mechanisms of cell regulation. It's interesting that a lot of these things we'll talk about in the upcoming videos are very common in eukaryotes and prokaryotes, and are very common across different living things. Cells communicate with each other via chemical signals. They can also respond or communicate with light signals and other types of messaging. But most of the time, this unit, we're going to be looking at chemical signals. For example, fight or flight, which you often hear about, is triggered by a signaling molecule called epinephrine. In your handout or your textbook, there is a picture of a cheetah chasing an impala. Sometimes we call this adrenaline. And you hear about that adrenaline rush when there's danger. Okay, you're either going to stay and you're going to fight, or you're going to try to run away. Okay, how do all those things like increased heart rate, increased breathing rate, the muscles getting charged for action, how do all of those things in your body, those thousands, millions of cells, all sort of like time that all together? How can they get that all together? And it's done through cell signaling. This adrenaline, this hormone, is used to coordinate that response. In the first section of the chapter, we talk about kind of an overview. External signals are converted to responses within the cell. So something is released by the cell, which causes a change in other cells. And in order to study this back in the 70s, we first started with microbes. Okay, microbes, single-celled, they tend to be simpler, and it gave us something to research to begin understanding how this happens. One of the first common things they looked at was Saccharomyces cerevisiae, which is the yeast cell that we often use to make alcoholic drinks, beer and wine and spirits. They noticed that there were two mating types, A and alpha. And the different mating types secreted factors specific to each type, and it's how the mating types found each other. So if we look at the picture here, okay, here's type A, okay, here's type B, or type alpha, and they're each releasing, okay, A will release an A factor, okay, which you see that the alpha has a receptor for that A factor, whereas the alpha is going to release an alpha factor, and the type A cell has a receptor that will accept that alpha factor. When they are close enough, they're releasing these little factors, these little chemical signals, and when they get close enough, it will actually trigger a response where they will grow towards each other and join together and fuse their nucleus and they'll join their genetics together. And so it's as they're kind of living out in nature, these factors kind of help them find each other. Once they're close enough, it will actually help them to grow together. So there's some kind of signaling done between these two types of cells to communicate in order to mate and create a new type of yeast cell. Now, how do those chemical messengers actually cause a response? We think there's what we call a signal transduction pathway. That's a series of steps that here it's kind of a complicated looking diagram down here, but you see on the cell surface here, there's all these different receptors here on the surface. Okay, and when there are different things, survival factors, hormones, growth factors, different receptors, when they receive a signal, you can see in here almost like dominoes. There's different pathways that will either go into the nucleus and change gene regulation, cause apoptosis or programmed cell death, cell proliferation, can cause the cell to actually form new cells or more cells, can cause the cells to grow towards or away from something. But you get all these little pathways where one thing, like dominoes falling, one thing hits the next, next, and it causes this cascade of molecules changing and will cause the cell response. Going back to prokaryotes or bacteria, cell signaling is critical among prokaryotes. One of the things we've noticed is that they use these chemicals to sense their own concentration, and they monitor their local density of bacteria, even bacteria of different species. We call this quorum sensing. A big example is when we go to the dentist and get the plaque cleaned off our teeth. That plaque is an example of a biofilm. Okay, a biofilm is formed by bacteria where they try, so they make a film of different peptides and stuff that will have the right properties to stick to your teeth so that more and more bacteria can build up on that biofilm. And eventually, some of the stuff they secrete, which can be acidic and stuff, will actually begin to cause holes in our enamel or dental caries or cavities. Example of this biofilm that we all deal with is on our teeth. If you've ever felt rotting leaves or something in a forest and they feel kind of slippery, have that slippery coating on it, that might be a biofilm of decomposing bacteria on that leaf, just trying to adhere to the surface, and they kind of work together to break down that leaf. In multicellular organisms, we can have what we call local and long distance signaling. So cells communicate, again, it's usually chemical. And when we talk about local signaling, animals and plant cells have different cell junctions, and we'll look at them in a second, in which adjacent cells, which means cells that are right beside each other, can communicate. And this can be from direct cell to cell contact. So in a tissue of cells where you have all these cells lined up here, okay, cell A here can communicate to cell B, which can communicate to cell D or cell C, and then to cell D, and so on and so forth. If the cells are side by side, they can communicate right through their cell membranes through cell junctions. Remember from previous chapters, here in animal cells, oftentimes that will be through what we call gap junctions. Junctions in plant cells, they have what are called plasmodesmata because they have this extra cell wall, which animal cells do not have. So these little openings called plasmodesmata allow the cytoplasm between the two cells to communicate with these little chemical signals right here. And sometimes it's on the surface of two different cells when they come in contact with each other. Cell-to-cell recognition. This is a big one when in Bio 2, when we talked about immune responses, and you have antibodies that recognize antigens. Okay, remember those were surface markers on the outside of the cell. That would be a type of local cell communication. And when there is a connection, when receptors hit a signal, it can cause a response. Okay, if this is the immune cell right here, and it finds a particular antigen right here, and says that's bad, we need to get rid of that. can cause this cell to either send out signals to other immune cells, or if it's a phagocytic cell, would actually devour this cell, would devour this cell and then try to destroy it. So again, looking at local versus long distance, a cell can signal to itself, which is called autocrine signaling. On some of the short distances ones that we talked about, this is often referred to as paracrine signaling. It's just they're either next to each other or they come in contact with each other. If it's a long distance signaling, oftentimes chemicals will travel through tissue fluids, lymph, or oftentimes the blood in the form of hormones. That would be referred to as endocrine signaling. And the ability of a cell to respond to that signal depends with whether they have the right receptors on their surface to respond to that signal or not. So even though it may travel throughout the plant's or animal's body, they will only affect the other tissues that have the receptors for that signal. Here on this slide, we can see some examples of that. Here's paracrine signaling, which would be considered local signaling, where there are, a bunch of here's the secreting cell here that's releasing the signal, the chemical signal messenger through the extracellular fluid, and when they hit target cells, will cause some kind of response. A specific type of local signaling or paracrine signaling is synaptic signaling, which you've studied before in the nervous system. Okay, when there's a neuron, the axon of a neuron here, and either another neuron here, or in the case of a motor neuron, this might be your muscle. They don't actually touch. Remember, there's the synaptic cleft between the two that the neurotransmitter goes across. In Bio 2, I know we talked about this with neuromuscular junctions, NMJs. There was a neurotransmitter called ACH or acetylcholine that was used as a signal when the brain is sending impulses to tell that muscle to contract. Over here, we see an endocrine cell. Let's say maybe this is in the adrenal gland. How does it pick up the heart rate? Well, it releases a signal oftentimes into the blood, and it travels through the body until it gets to the heart, where it will cause a response. So it's traveling a longer distance using these hormones. Again, an example of endocrine signaling. Okay. The actual process of that signaling, a lot of the early research was done by Earl W. Sutherland in 1971. Actually won a Nobel Prize based on what he found. And he suggested, and that we still think today, there's basically three main steps. Number one, the cell has to receive the message. Number two, there's transduction of that message into the cell. Throughout the cell in order to, number three, cause a response of some kind. Here you can kind of see an overview of that response. Okay, so number one, we have reception. So here's the cell membrane here. And then we've got some type of receptor on that cell membrane. Here comes a signaling molecule like a hormone, like adrenaline. When it hits a target cell, this would be the target cell here for that signal. It would go through, once the receptor hits that, it would cause the second part, transduction, to take place. And this was that big complicated diagram I showed you earlier where there were all those different little pathways, where there are relay molecules, and sometimes it might just be one. Sometimes there's several different pathways and several different molecules as it travels through the cell to cause a response of some kind, whether that's apoptosis or some genetic response of some kind of other hormone or protein, or even causing the cell to divide and make cell proliferation to cause more cells. So cell communication, we talk about reception, transduction, and response of that signal. In the next couple of videos, we want to take a closer look at each one of these steps in each of those videos.